Anyway, um, if you're following along in your notes, your DNA notes look like this. And the way that I count it for in-person folks, I'm calling this like page one, and this right here like page two. So like page one and two, since they're kind of shrunk down. Thanks, Kevin. They're on the actual like same physical piece of paper. So if you're following along, you'd be on like page one, two, three, four, five, six, seven. You'd be on like page eight. Up at the top, it says AP Biology Regulation of Gene Expression. And for you guys at home, I'll show you what it looks like. It's the page that looks like this. So for you guys, it's a right-hand side of the page. And up at the top of that right-hand side of the page, it says AP Biology Regulation of Gene Expression. And if you're printing it off like a true one page at a time, it'd be like page eight. And in the middle of the page, you see a bunch of like kind of Lego block looking things that looks something like this and this. That's like, there's like three pictures of things that look like that on there. Okay. Um, so first off, have we looked at any of this glowing stuff in here? Okay, I didn't think so in third period. So just a real quick introduction to gene regulation stuff is actually really cool. So this is a jellyfish, and this jellyfish glows. And so what we find is that, wait, who did I just buzz in? Mercy. Mercy, let me know if you, Mercy, let me know if you need to take the quiz. So if you took it. Quiz. I just came back from a dentist appointment, so okay. Then I take it, but I finished it. Okay, all right, cool. I was just making sure. Some people are like in and out because of internet connections, and I'm like, I don't know if I need to like, if this is your first time in class or not. So cool, thanks. So we got this jellyfish, and he glows blue. And so scientists are like, all right, well, if it glows blue, there's probably a protein that makes it glow blue, and there is. Well, if there's a protein that makes it glow blue, then that means there's some DNA, some genetic code that codes for glowing blue, and there is. Okay, well then if the genetic code is universal, and I can find that code of DNA that says glow blue, I can splice that into any living thing, and I can make any living thing glow blue. So that's what we do. So out in nature, we found all these different colors. All of these are proteins that we found in nature. Most of them are like deep water fish and jellyfish and stuff like that. So these are all different colors. So somewhere out in nature, there's a thing that glows green, like there's a jellyfish that glows green. And so we have the DNA isolated for the protein that glows green. So we've isolated all of these. And then we can splice them into any living thing that we want, like a moth. All the other moths are jealous. He says it sucks. He's like, well, I'm blind when the lights are on. That's all I can see. Yeah, you can do it to any living thing. Now, the reason we don't do it to humans is, first of all, what's the benefit of having like glow in the dark skin or eyes or something? And then second, we do this like in a test tube. So we're taking like moth sperm and egg, or like the first few cells, like as they're developing, and we have to like splice it in then. Otherwise, like I'm already done. I'm 80, 90 trillion cells. So if you go in and like tweak one of my cells in my skin, like is my skin glowing everywhere? No, you'd have to like do a little mini operation on every single skin cell to inject that DNA, right? So the only way to make it to where my skin glows is like when sperm and egg meet in a test tube, you stop it, go in and change the gene, and now that original cell that becomes me turns into 2, 4, 8, and it carries on the green gene. So we had to do, this was a test tube baby moth. Do you think in the process we accidentally killed some moths? Yeah. So it might be like, hey, we did this to 50 moths. 49 of them, we screwed something up. But one of them lived and his eyes glow. Well, obviously with humans, you wouldn't do that. There's no benefit. And you just sacrificed a bunch of humans so that you can have like Somebody that looks cool when the lights are out and a black light is on. So we made moth eyes glow. Here's some mice. The controls do not glow in the picture. And then here, let me turn the volume down. I need to remember to turn the volume back up so that I can hear you guys at home in case you need something, or in case you have a comment, or in case you're asking a question. But here's a mouse so that you can see that the things are still alive. So this mouse actually is like a normal mouse, but if you put a black light on, he glows green. He took a little pee there. We also have mice that are hairless so that you can see that their skin glows. Kind of like those cats. I think they're Sphinx cats. The ones that look like prunes, only they're animals. People are like, oh, it's so cute, it has no fur. And you're like, that is a bald cat. That's ugly as heck. Then here's one last example. I clicked play. Let me click play again. There we go. This is an axolotl. Axolotls are basically, they're like a type of amphibian, so they're kind of like a salamander. They have external gills, so right behind where their ears would be, when you see those kind of tree branch, almost like pine needle tree branch looking things, those are their gills. They have external gills. You're used to fish with internal gills, so they go like this to like pull water past their gills. These got the blue fluorescent protein instead of the green. So when we shine a light on them, they fluoresce blue. When the black light's not on them, everything's the same. So their skin's the same. Everything works the same. All we did was whatever proteins are in there, we made them glow. And so my favorite lab that we do in here, we just finished my second favorite AP Bio lab, but my favorite lab in here is when we take a gene from green glowing jellyfish and splice it into bacteria, E. coli bacteria, and we make some glow in the dark E. coli bacteria. We also make them antibiotic resistant, so like wash your hands. Like E. coli is kind of a big deal, right? Don't they pull like about every 2 or 3 years they'll find out that romaine lettuce is contaminated with E. coli, and like they pull all the romaine lettuce off the shelves, and there's no Caesar salad anywhere. And so we're like, hey, let's take some of that, let's make it antibiotic resistant, and then play with it. That's what we do. Okay, so that's some of the interesting stuff that we can do with gene regulation. But of course, now we want to know, how does all this stuff work, Mr. Cotton? So let's start off with this. And this really isn't in a certain location in your notes. So if you're like, where would I write this? Or is there a diagram or someplace I could put it? Not really. It is very important. So you do want to just find a space to like write this or draw the picture or take a picture of it or whatever. It is important to understand this, but I don't really have it in your notes anywhere. It's called negative feedback. So this explains like, okay, let's say that we're trying to figure out what would happen if we eradicated malaria carrying mosquitoes. So for example, there's some mosquitoes that live around the equator. We study this a little bit more in evolution, but there's some mosquitoes that live around the equator and they carry malaria. We have mosquitoes in Dallas, Fort Worth, but they cannot carry malaria. So that's why we don't really hear much about malaria up here. You've heard about it in like books, but like it's not like your friend's absent from school because you got malaria. So, but start heading south, like Central America, parts of Mexico, on into Brazil, kind of where the equator is, and those are common. So, what would happen if I like eliminated? I just somehow eradicated this one species of mosquito from the earth. They're like, well, that would be great. The number 8 killer of people in third world countries is malaria. Like in the year 2021, I know we're only like a month in, but like by the time this year's over, or we'll just look at last year, the data's not all in, but when it is, the number 8 cause of death, number 9 cause of death, somewhere around there is going to be malaria in third world countries, not in the US. Third world countries, around the equator. Okay, so you would think they'd be fine, but you've seen food webs, right? All of a sudden, I destroy this one mosquito, and then that means this particular type of frog goes extinct, and then like the snake that eats that frog, and before you know it, we eradicate mosquitoes near the equator, and like polar bears go extinct or something. You know what I'm talking about? So it gets really complicated. When we look at a big food web, like a realistic food web, it's like incomprehensible. So what we do is we take a small chunk of it and we're like, here's a food chain. A eats B, B eats C, C eats D. What happened if B goes extinct? Well, C and D go extinct. Very easy to understand. That's what we're going to do. When you're like, okay, Mr. Cotton, previously you would have a picture over here and you'd have like you'd have a gene. Oops, clicked the wrong one. You'd have a gene. So nucleus, perfectly drawn circle, by the way. Histone, 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 histone. Other strand of DNA. I'd have a promoter and a terminator. And I'd be like, all right, this promoter's on because transcription factors land there. Down here, there's a promoter and a terminator. This promoter's off. There's some sort of protein like blocking the way on the promoter. So that's how I know to read this gene because it's on and don't read this one because it's off. But then you're like, okay, well, like, how did that protein know to land there and turn that one off? Or how did those transcription factors know to land there and turn that gene on? Like at some point, like, who's doing the thinking? When my cells are like running themselves and managing themselves, like my body's managing its own blood sugar. My body is managing my temperature. My temperature is, I run kind of low, so I'm like in the high 97s. So my baseline body temperature is like 97.8. And it's within half a degree of that, whether it's 90 outside or whether it's 40 outside. So how does my body manage all that? How does it know? Like, oh, it's getting too low, shiver. Oh, it's getting too hot, sweat. Like, how does it monitor? Like, keep sweating or stop sweating? How does it like run itself? Like, who's doing the thinking? If transcription factors land there, well, how did they know to land there? Well, in reality, it's complicated like a real food web, but we're going to kind of look at a small chunk. So we're going to watch a small piece of DNA regulate itself. You'll be able to understand that, and then you're like, okay, now take that, kind of like here's how a food chain works. Now imagine I've got 50 billion food chains, and yeah, it's complicated, but you can at least kind of see how it might work. So that's what we're going to do. Okay, so let's say that I have, that's the camera I want to be on. Let's say that I want to make 100 tryptophans. So there's this amino acid called tryptophan, TERP. That's the official abbreviation. You don't have to know the name of it or anything, but tryptophan. My body can make them. So a little bit over half of the amino acids, there's 20, and about a dozen of them my body can just make. So if I eat nothing but like just table sugar, then my body can manufacture about 12 of the amino acids. But 8 of the amino acids my body can't make. That's why if I eat nothing but sugar, I will die, because I can't make some of the amino acids. They are essential nutrients. If you ever hear of something being an essential fat or an essential nutrient, that means you have to eat it. Your body can't make it. Vitamin C is essential because I can't make it. My dog can. He has a gene for making vitamin C. I have that same gene, but it's got a mutation. So we have broken vitamin C making genes. So if we want vitamin C, we have to eat it. Vitamin D is not essential. If I go outside and the sun hits my skin, my body can manufacture vitamin D. I might get skin cancer in the process, but my body can make it. Okay, so my body can make tryptophan. Let's say that I always want 100 tryptophans, just like I always want my body temperature to be like 98, 99. I always want my blood sugar to be like 90 deciliters per milliliter. I always want to have 100 tryptophans around, about. I don't want 200, and I don't want 6. I want about 100. So what if I have a diagram like this? And again, anytime if you ever need something, you can always take a picture. If your camera kind of goes up and does this thing, I'll make sure I'm out of the way. So here's how this works. I start off with this one chemical, and they represent it with a rectangle here. So that's going to be this. Then I have this enzyme number 1, and it tweaks this. Then enzyme number 2 grabs it and changes the shape again. Then enzyme 3, enzyme 4, enzyme 5. Spot on. Then it looks exactly like this. So I just made tryptophan. Number 5 down there, that is tryptophan. Okay, but I only want 100. 200 is bad. 50 is bad. I want more or less 100. So what could I do? Well, what if I have like 100 of enzyme 1? So here's enzyme number 1. And again, what we're learning right now is basically when it seems like cells are thinking and making decisions like, again, my body is managing its own temperature, its own blood sugar, all of this, like how are my cells thinking? This is how we're thinking. Okay, so I have this enzyme. That's the active site. Let me throw this out there for the Zoom crew. So Zoom folks, you get first dibs at this. That's the active site. So what's this called? This would have been Unit 3. We might have introduced it in Unit 1. What's that called? This whole thing is an enzyme. That spot's my active site, where my substrate goes. What's that called? Or, if you don't quite remember what it's called, what goes there? Nope, you can submit it now. It won't hurt you. Good, thank you, Mercy. It's an inhibitor. So Jason, to answer your question, you can submit it now. It's cool. If you ever had like 1.5 voluntary homeworks or something, I just kind of keep track of the half. And then when you add another half, I give you another 100. I wouldn't like put in, oh, you got a 100 and a 50. That'd be horrible. I'm penalizing you for doing stuff kind of thing. So yes, Mercy, an inhibitor. And then does anybody in person remember what this site for the inhibitor is called? It had a specific name. It starts with an A. Allosteric. Was that Kevin? Okay, an allosteric site. So this allosteric site can bind an inhibitor. So say my inhibitor is shaped like this. So I'll put an I for inhibitor. Whenever that inhibitor goes into that allosteric site, allo means different. So it literally translates as different site. It's different from the active site. So whenever that inhibitor goes in there, my active site changes shape. And when it does, that enzyme is off. My substrate can no longer fit in there. What happens whenever I take that inhibitor away? Yeah, it goes back. So it's a reversible change. What was that called? I'll throw this out for Zoom first also. What was that called when a protein has a reversible change? Not denature. Denature tends to imply, it's technically not always the case, but it tends to imply like ruined and irreversible. But what was it called when a protein did a shape change? Again, the Zoom crew gets first dibs. I am a teacher of the people. I include my Zoom students. No, it's not that, Reagan. Thank you, though. Anybody else? I've got a protein and it changes shape, but it's reversible, so it can change. It can change forward, change back, change forward, change back. Generally, a permanent, irreversible change we generally call a denaturation. All right, anybody in person? It's like 2 words, a something change. So I guess it's 1 word. Yeah. Good. Conformational. I conform to a different shape. The example I would have given like however many months ago would have been, nobody looks like they have a super out of control backpack. So let's say you've got like a super heavy backpack. You've got a lit book. Lit books are the biggest books that you never read, right? Like here's 1,800 pages. Time for Canterbury Tales. Let's read the Nun's Tale, and that's it. So you read like 12 pages out of that book. It's that thick. So here's how I stand without a backpack. Put like 40 pounds in a pack on my back, and how do I stand? I hunch forward. That's my most comfortable state. Take off the backpack. There you go. Put the backpack on. There you go. That's like a conformational change. So this enzyme changes shape whenever I add the inhibitor, add the backpack, and take it away. Okay, so let's say I've got this enzyme, 100 enzymes, and their job is they make tryptophan. So again, maybe it's enzyme number 1 or 2 or 3 in this chain of enzymes that makes tryptophan. Okay, this is a very hard question. If I want to always have 100 tryptophans, good, I now see that in the chat. Sorry about that. You guys might have typed it and maybe things are just a little bit slow, but yes, conformational change, which is a non-competitive site. That's true. It's a type of non-competitive inhibition. So thank you guys for throwing answers in the chat. Okay, so I've got 100 enzymes and they make tryptophan. How could I get this system to regulate itself and always have about 100 tryptophans? As is, it's a very hard question. If I give you hints, it might be a little bit easier, but is there a way with just chemistry for this thing to be like, oh, well, if this were true, then you would always make 100 tryptophans. Anytime you drop down to 99 tryptophans, you would make another one. And all the basic ideas you need are like up there on the board. You don't have to pull something else out of your brain from 2 months ago or something. So like when I explain it, I'll just be like manipulating stuff on this board right here. I want to always have 100 tryptophans. When I use tryptophans and I get down to like 99, I want to immediately somehow know to like make one more, not 100 more, one more. What could maybe that inhibitor be? Yeah, it's hard. Some of y'all are like, I don't even know what's going on right now. Like he's asking questions, but like I don't even know. Like I don't even have any idea. That's an enzyme. Like I have no freaking clue what he's looking for. That's true. So I see a few answers in the chat. So whenever the inhibitor enters, that enzyme will change shape and it'll be off. So that's true. That's one thing. And the other one, does the inhibitor get removed to make more? That's true. So when I remove this inhibitor, won't this enzyme start making more tryptophan? I have 100 enzymes. What's the identity of this inhibitor? Tryptophan. So I have a machine that makes tryptophan. You know what turns off the machine? Tryptophan. Let me show you what I mean. I've got an enzyme. I've got 100 of them. So they're all making tryptophan. Let's say that they make 90 tryptophans. My inhibitor here is now tryptophan. So that's what will turn off this enzyme. So let's say I've made 80 tryptophans. What should, in theory anyway, I realize we're like relying on random collisions, but what should those 80 tryptophans be doing once they're made? They should be turning off the enzymes, right? So how many enzymes should, in theory, in a perfect world, how many enzymes will be off if I have 80 tryptophans around? 80. So how many enzymes will be on? 20. So will I still be overall making more tryptophans? Sure, 20 enzymes are still working. All right, let's say I get to 99 tryptophans. All right, that means 99 enzymes should be off. One enzyme is still on. What is that enzyme doing? Making tryptophan. What happens when I get to 100 tryptophans? Yeah, they should all be off, right? There should be no enzymes that are on. So when I get to 100 tryptophans, is anybody going to make tryptophan number 101, 102, 103? No, so I stop. So every enzyme is inhibited by tryptophan. Now, let's say that my body wants to build insulin. Insulin ends in IN, so insulin's a protein. Maybe when I'm building insulin, I've got this chain of amino acids, and like amino acid number 12 is tryptophan. So when my body needs to use a tryptophan, it's going to take one from that enzyme, right? That tryptophan was turning off the enzyme, but I just removed that tryptophan to build insulin. So now how many tryptophans are there? Only 99. So doesn't that mean that one of my enzymes is back on? And what will that enzyme do? Make tryptophan. So now how many tryptophans will I have? 100. Until I use one. Once I use one of these enzymes will turn back on. So I'll have 99. I'll turn back on the enzyme. I'll have 100. Now, don't I always have 99 or 100 tryptophans? Doesn't it seem like it's intelligent and like somehow the enzyme knows when to make more tryptophan and it monitors how much there is? But is it really intelligence or is it just chemistry? Like it's just chemistry. That's what you are. Now, expand that to every other enzyme in your body, every other chemical reaction. We just learned a food chain. Now try to picture that on the level of like the world's food web. And that is you. So you're just a bunch of enzymes and inhibitors. That is your essence. It's not very romantic, especially for like Valentine's month. I love her. I love her essence. Just a bunch of inhibitors and chemistry. That's what you love. I love her personality. Yeah, that's like 80% DNA. You just love the order of A's, T's, G's, and C's. So, okay. So now let's look at the genetic version. We were looking at like turning proteins on and off because that's a little bit easier to understand. Now we're going to look at this in bacteria. So right here, I have gene 1, it makes enzyme 1. Gene 2, it makes enzyme 2. So I have 1, 2, 3, 4, 5 genes. Well, in bacteria genetics is a little bit simpler than in us. So we'll study bacteria first. In that picture that was just up there, we had gene like 1, 2, 3, 4, and 5 that ran the 5 chemical reactions. Those are these genes here. So what we were calling 1, 2, 3, 4, 5 before, over here are called A, B, C, D, E. So if I click back, gene 1, 2, 3, 4, 5, I'm just calling it A, B, C, D, E. So in bacteria, these are those genes, A, B, C, D, E, 1, 2, 3, 4, 5. Okay, so when I look up here, some parts of this should be familiar. I've got a promoter. RNA polymerase is still the guy landing on a promoter. So we had that in our picture. Again, my nucleus over here, we had like promoters and termination sequences. So that part's the same. This actual word is new. The concept is not new, but this word is new, the operator. The operator is the name of the on-off switch in bacteria. In eukaryotic cells, we don't call it an operator. We call it a regulatory sequence. But their job is the same. So it's almost like, I don't know, if you ever work on a PC and you're typing crap and then you go to like an Apple, all of a sudden everything's like on the opposite side. It's like, I'm trying to close the window. It's like, the close is on the left. Trying to open this. And you're like, well, and a Mac, it's on the left instead of the right because it's different. So same thing. An operator is like a regulatory sequence in us. An operator is an on-off switch. Okay, so a couple of questions. Do you think, and this is for everybody, So if you're in person, you can answer. If you're online in the chat, you can answer. And just like always, when I'm looking like this, that's me looking at the chat. So a couple of questions. Question one is, right now, as is this gene in the on position or the off position? And then, actually, I'll start with that one. So do you think that as is up here, this thing is currently on or off? Like, am I making tryptophan? Or is it currently in the off position? And we'll vote here in a second for you guys in person. And I'll wait a little bit longer. I know that the chat's a little bit laggy. Maybe haps. I like that. Yes, I won't announce what your answer is, but it is correct. It is correct, maybe haps. All right. So if you're in person, if you're online, you can still put yours in the chat because it's going to be a while before you understand what the in-person students say. So raise your hand if you think it's currently in the on position. That gene's on. Raise your hand if you think it's off. Okay, it's on. What evidence is there that it's on? There's kind of two things. One thing's a little bit tricky, and then the other one's like, oh, yeah, I guess that does mean that it's probably on. Kevin. Yeah. So one thing is like, well, it looks like I'm reading it and maybe making the piece of RNA, but maybe that just says what I'm supposed to do. So I'm kind of uncertain there. But I have this thing right here called a repressor, and it's inactive. So that means this is probably on, right? Like if I have something called a repressor, that means stop. And if currently the stopping mechanism isn't active, then it's like, okay, well, I'm probably on. So it's on. Good. Thanks for you guys that put things in the chat. So it's inactive. All right. Take a guess. Let's say I wanted to turn this off. What would change in my drawing? Like I'm getting ready to click and show you another picture of like, oh, here's what it's like when it's on. What's that picture going to look like? And this is for anybody, so online, in person, whatever. What will look different in my picture when I want to turn this off? It's currently in the on position. Will my repressor need to become active? Yeah. So what will it look like in the active state? What will look different? Yeah, maybe it can attach. Where would it attach? To the operator. So right now, why can it currently not attach to the operator? What's not quite right? Yeah, the shape. The shape is kind of like this, right? So what do you think happens to the shape of the repressor when it's gonna fit the operator? Now instead of being shaped like this, kind of more V-like, now it's more like field goal posts or rock and roll. So this will have to change shape like that. These genes all eventually make tryptophan. So what's going to cause this guy to change shape to where it fits in the operator? What's going to turn off my tryptophan genes? Tryptophan. Tryptophan's going to bind right there. This guy will change shape. He'll fit into that operator. And now my RNA polymerase, he'll either be blocked, like if I have this giant chunk sitting in there, he'll either be blocked to where he can't land, or he can land, but then he's like stuck. You can kind of think of it as like this is train track, and RNA polymerase is the train. If I like drop some giant like 10-ton thing on the train track right there, then like the train can't get through. So my repressor is going to physically block. It's going to act like a giant elephant on the train tracks. It's going to physically block RNA polymerase. So when we click forward, that's exactly what we find. Here's tryptophan. And whenever tryptophan hits my repressor changes shape. Now my repressor can do some repressing. He'll fit into that operator. And once he does, I've turned the system off, which kind of makes sense. If I'm a heater, like this, I'm sure you can imagine a heater, but I'll do this anyway. Yes, so to turn it off, the repressor is attached to the operator. Good. Change the formation. Yeah, the co-repressor would hit and like change the shape of the repressor. Yeah, good. So over here is like a little heater thing, and it currently says 73. Let's say that I wanted to warm it up. I wanted it to be 75. And pretend that this thing actually makes the heat. I know that like this is just the controller, and the thing that makes the heat is probably like up on the roof somewhere, some giant thing, some sort of compressor or whatever the hell. I don't know. There's probably some U joints in there, sockets. I don't know. So let's say that this thing also makes the heat. So I set it at 74. So it's going to start making heat. When it gets to 73, it's not there yet. So it's going to keep making heat. When it gets to 74, how is it going to know to turn off? Well, it's making heat. And what tells it to quit making heat? Heat, right? So same thing. That's what's happening here. When I'm making tryptophan, what tells me to quit making tryptophan? Well, tryptophan. I call that cinematography right there, in case you were curious. Okay, so now let's go back to this. They call this whole process negative feedback. Can somebody give me a logical reason why I would call this, whether I'm going here and turning off a protein, or whether I'm going here like we just did and I'm turning off a gene? Can anybody give me a logical reason why you'd be like, okay, I can see why they call that negative feedback regulation. I can see why they call that a negative feedback loop. Anybody got an idea? Are you thinking about it, Aiden? You look like you're gonna maybe. Okay, why do we call it a feedback loop? Yeah, so like feedback is, I'm kind of going back, I'm giving feedback. Like in English class, you might edit each other's stuff. So I write some sort of essay and then I take it to my group and they kind of give me some feedback. They kind of go back and feedback information to me and I rewrite it. Well, this is negative feedback because I'm feeding back and I'm saying, stop it. What would positive feedback look like then? Yeah, feedback and like keep going. And if I say keep going, then you go faster and faster and faster and faster and faster. That's positive feedback. So what we find in the world of biology, life, is that as a general rule, if you ever see something that's a steady state, that's probably governed by a negative feedback loop. My blood sugar. If I chug a 2-liter Coke and eat 3 Snickers, my blood sugar is going to start to go up. Insulin will bring it down. If I don't eat for 2 days, then my body's going to start breaking down myself to bring my blood sugar back up. So I keep my blood sugar steady. My body temperature, steady. My blood pH, steady. I try to keep a lot of things steady. Those are usually governed by negative feedback loops. Positive feedback loops look like this. Once you start something, it just goes faster and faster and faster and faster. It's exponential growth. So this is more of a positive feedback loop. Thinking of negative feedback is easy. Like I said, blood sugar, body temperature. What about positive feedback? Uh-huh. Blood clotting. There's never really a time. I always think of two. Blood clotting is one of them. So there's never a time in life where I sever a vessel and I start blood clotting and it's like, hey, slow down on that blood clotting. No, it's always like, hey, plug that hole as fast as you can. So in blood clotting, chemicals activate chemicals, activate chemicals, go, 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 go. Does anybody know the other one that's commonly mentioned in books? childbirth. So, personal story. So let's say you're watching a show. Maybe you're in some K-Cow classes and you get to go to hospitals and stuff like that, or maybe you get to go to a vet clinic. I don't know that they let you see like human birth, but you could probably see like, puppy birth or something. If you're at the vet clinic and there's a dog giving birth, I'm sure they'd be like, hey, you want to see it? So has anybody ever seen live birth? Maybe you had your own cat or dog or something at home. Okay, but you've seen television. Have you ever heard them say like, she's dilated this many centimeters? And you're like, so what exactly is dilated? Like I know dilated means like big, but like what exactly anatomically is dilated? Because I'm very concerned here. Well, here's what's dilated. So it's at first you're like, oh, I know that. That's the beginning of a balloon or a uterus. So this is a uterus. When you were a baby, you were growing inside of mom's uterus. So here you are. You're so cute and disproportionate. You're hanging out in mom's uterus. Now, as you get bigger and bigger and bigger, eventually that applies more pressure on the uterus. The uterus is a giant muscular sac, kind of like your stomach is a giant muscular sac, except this one makes babies instead of eating stuff. So, or it allows the baby to grow. It doesn't make babies. Uteruses don't make babies. I want to retract that. Mr. Cotton said uteruses make babies. So here's a uterus, and the opening to the uterus is called a cervix. Well, when a woman is giving birth, like that cervix just gets bigger and bigger and bigger. So when they say she's dilated 8 centimeters, that means that her cervix is that open. And when the doctor looks up in there, that's what he sees is like that kid's head. I'll be like, he has dark hair. And they're like, how do you know? And it's like, I'm looking at it. Like, I can see his head. He has dark hair. So usually around 10 centimeters is when they're going to like tell her like, all right, you're giving birth. Now it's time to push. That's 4 inches in diameter. That means her cervix is that dilated. Isn't that crazy to think that like a doctor's looking up in there and they're like, I can see the head. And it's like, wait, he means he can actually like he could touch that kid. Isn't that weird? I think it's weird. So here's how positive feedback works. You start, and that uterus gives like a small contraction. So, like I said, personal story and a life lesson to males. So, you always hear stuff. You'll always hear, like, if a woman, let's say that let's say you're a male, you marry a woman, and she's, the classic, like, do these pants make my butt look fat or whatever? And, like, common knowledge is you say, like, well, no. But sometimes you're like, my wife and I, we're tight. She has a sense of humor. And so you can like make jokes or whatever. And so you make a joke and you're usually right. And so I tend to do things like that. So wife's pregnant and she's like, we're at home and she's like, I think I just had a contraction. And so at that moment, I knew to joke or not to joke. Common wisdom says, don't joke. She's pregnant. I know what you're thinking, but don't say it. It's not funny. The other side is like, this is my wife, Jessica. I know her. She knows me. She'll think it's funny. One of the reasons she likes me is because Alan Cotton would say a joke right now, and he's going to make it seem offensive. So that's what I went with. And I was like, how can I make this as funny as possible? Oh, I'll refer to women as y'all. Y'all always talk about like pregnancy as being all painful and stuff, and you're not even sure if you just heard it, if you just felt a contraction. And she kind of looks at me, and I'm like, LOL? And that didn't help. So later on, I was like, I was just joking, and that didn't help either. So what happens is you get like a small contraction. Well, that pushes the baby like more against the cervix, right? If I've got a water balloon and I've got something in there and I kind of squeeze like that, then aren't I like pushing something out of the water balloon. So that pressure is part of a positive feedback loop. So every time that baby's head gets squished against the cervix, it feeds back and it tells the uterus to push harder. So she has a contraction. 40 minutes later, she has a contraction. 35 minutes later, she has a contraction. 30 minutes later, 25, 20, and so forth. Every time she has a contraction, they get more intense. So it's kind of like your uterus goes like, like, hello. And you're like, did that just say like hello? And then it's like, hello. And then like eventually it's like contracting more often and like with more intensity. So when her contractions are like 20 minutes apart, she's like, yeah, I'm gonna. Okay, that contraction's over. What were we talking about? And then by the time the contractions are just like when you're giving birth, eventually your uterus is just like, like screaming like, ah, like that. And then out comes the baby. What's positive feedback? Once you start, more often, more often, more intense, more intense, more intense, go, 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 go. Positive feedback. And a life lesson. Okay, now let's go back to this. This gene's natural state is the off position. That makes sense. So once I start making something like tryptophan, then I'm sorry, it's naturally in the on position. So once I start making something like tryptophan, then eventually it'll be turned off. Okay, but now, what if I take like your desk? Let's not use your desk. I clean those a lot. Pretend that it's 2 years ago before COVID. Isn't there bacteria all over your desk? There's not now because after every class, we go around and we wipe down your desk. So this school is as clean as it's ever been. But even right now, aren't there bacteria floating around in the air and like falling on your desk? So why are they not growing so well that you can see like colonies? Like in microbiology lab, I can literally see like colonies of bacteria with the naked eye growing. Why can't I see colonies growing on this countertop? What's limiting their growth? I mean, there's like tons of things that could be, but in this situation, if I dumped bacteria on this countertop right here, why, when I come back tomorrow, is there not just like a whole layer of funk growing there? What are they missing? Yeah, nothing to eat, no food. So what if I dumped some like chicken broth on here, left it, came back a day or two later? Wouldn't it be covered with nastiness? Because now they have food, so now there's like fungus and bacteria growing like crazy. Okay, so if I'm a bacteria, I can make an enzyme called lactase. What does lactase break down? Yeah, dairy, lactose, sugar. So I can make lactase enzyme to break down lactose sugar, and lactose sugar is in dairy. So anything made with milk, so cheese and all that stuff. Okay, now, if I'm a bacteria, I have to like only make lactase when I need it. Doesn't it take energy probably to make lactase enzymes? Sure. So since it's life or death, like me conserving my energy, shouldn't I only make lactase if I like fall into a glass of milk or something, or land on some cheese? If I'm a bacteria and I land on this tabletop right here and there's no lactose, it's a waste of energy to make lactase. So here's what that means. When it comes to making lac, the lac operon, don't worry about the word operon at the moment. When it comes to lac, lac is naturally in the off position. So this is when we make lactase genes. Well, naturally, they're off. What's going to turn on lactase genes? Lactose. So in this picture up here, what shape is lactose? How could I turn this gene? What do I have to do to turn this gene on? What do I have to change in the picture? Right now it's off. Yeah, I gotta remove the repressor. So I've got to get it to maybe somehow change shape. What might make it change shape? Lactose. What shape is lactose? Yeah, looks roundish. At least part of it's round. So when lactose fits in there, this guy changes shape, falls out, and now I start making lactase. So now I only make lactase when lactose is around. So this is how bacteria think. How does that bacteria know to not waste energy making lactase? But when he falls into a cup of milk, he knows to somehow start making lactase. He doesn't know anything. Lactose removes the repressor. And so now it's all chemistry. The decision is made. All of these things that we're calling decisions is just a bunch of chemistry. Right? Even your psychology. You're depressed. You're like, well, it's part of my personality. We can give you Zoloft, and we just changed your personality with a pill. Like you view Monday differently when you wake up. If you used to be depressed and not on antidepressants, and now you're on antidepressants, right? And all we did was change a chemical in your brain and it changed your personality. You're just a bunch of chemical reactions. So it's Valentine's Day soon. At your age, you probably, if you have a Valentine, you probably haven't been with that person, for like years, like I have, right? I've been married for about as long as you guys have been alive. So at some point, as you get older, it's Valentine's Day. You're like, all right, what I'm going to tell her. I love her. I love you. Okay, I've done that. Your face is like a sunset or something. And the moon and the planets are there because of you. So eventually, you give roses, you compare it to flowers, sunsets, and beauty and all that. Then you're like, all right, my wife and I met in 2002. I'm like, this is the 19th Valentine's Day coming up. I'm running out of ideas. I love the chemical reactions that happen in your brain that make you who you are. Happy Valentine's Day. That one's a winner. That's what my wife's getting this year. Here's what's funny. I have literally written something almost like that in a Valentine before. When we get to the nervous system unit, the way that you generate electricity in a nerve cell, which is a thought, is sodium and potassium moving in and out of nerve cells. So I wrote something about how I kind of started it off sort of poetic sounding, like I was going for the sunset. And she's like, you've compared me to a sunset before. I went that way. And then I was like, I love the way the sodium potassium moves in and out of the voltage gated channels in the sodium potassium pumps to make you who you are. Happy Valentine's Day. And I actually wrote that because I know my wife, and that's funny. However, when she was pregnant, I was wrong. That was not the time to joke. Anyway, you can use that if you want on your Valentines. Okay. So the next thing is let's look at like humans. Eukaryotic cells. Eukaryotic cells are different. See, you get biology, you get knowledge, you learn how cells work, romance, I mean, unstoppable. I'm just helping you guys like crazy. So you might remember we had DNA wrapped around histones. If you're following along in your notes, flip over to the next page after all that bacteria stuff. And now the page looks like this. You've got a histone that looks a little bit like the one on the screen up there. The page looks like that. At the very top, it'll be a left-hand page for you guys. It says Gene Regulation Eukaryotes Concept 11.3. So it's that page. And right here is a histone with DNA wrapped around it. Okay, so I've got DNA wrapped around a histone. If you happen to come across the word nucleosome, that's just what they call DNA plus histones together. Like, I don't know why. What's that? DNA? What's that? A histone? What do we call it when they're together? A nucleosome. All right, fine. So here's where a lot of this stuff about gene regulation gets really interesting. This is where you learn stuff that you probably didn't really learn in ninth grade about genetics, and it changes the way that you view life. So I've got this histone. Here, it looks like the histone is like really tightly packing down this DNA. You might remember, if I grab my roll of histones, that's what I call them at my house. You, hey, love, can you hand me the roll of histones? My poor son, he'll go to somebody's house. You know, he's like 7 or something, and it's the first time he gets to spend the night at somebody's house. He'll be like, we're having pizza. It's like, can you hand me a piece of histone? And they're like, Cotton, what the hell's wrong with your kid? And I'm like, well, I call paper towels histones. Probably should have thought of how that's going to screw up my kid. But he'll get that question right on the test in 9 years. So here's my roll of histones. Histones have positive charges. Histones are proteins, and some amino acids are positive. So I just have this chain of positive charges on histones. And then over here's my DNA. See, it's already tangled in a knot because I didn't have it wrapped around histones. Okay, so here's my DNA. And my DNA, let's say it's all double helix together. If I want to read it, like transcription and translation, I've got to be able to like unzip it, right? I got to be able to like find a promoter, unzip it, start making RNA. I have to unzip it. Well, my DNA can get tangled. So in order to have my DNA not get tangled, I have it wrap around histones. Histones are positive. The phosphate backbone in DNA is negative, so they attract. But now, while my DNA is wrapped around histones, like this picture over here, can I unzip it very easily if it's like tightly wrapped around histones? No, so when my DNA is packed around histones, those genes are off. The first step to turning on a gene in eukaryotes, prokaryotes don't have histones, bacteria don't have histones. We do, plants do, fungus does, protists do. So eukaryotic cells do. The first step in turning on a gene is I've got to get those histones and those strands of DNA to let go of each other. I need to get the histones to like loosen up its grip, maybe like that. So now, put that behind me. So now my DNA is kind of hanging off. Now I can unzip and read this part of DNA. Okay, so what would I have to do to get histones to let go of DNA? I'll throw that out there for my Zoom crew first. So I'll repeat it. If you're on Zoom, give it a shot. What would I have to do to get my histones and my DNA to like let go of each other? Like what would be a logical thing I could do chemistry-wise to get them to let go? I want to turn this gene on, so I have to somehow get histones and DNA to let go of each other so that I can unzip. What might I do? Yep, I got to break some bonds. So right now there's a positive and a negative. What else could I do? So I have to get them to, I have to interfere with these bonds somehow. If I break them, they might like remake right away. You're on the right track though. What do you guys think in person? What could I do to this paper towel roll to get it to let go of DNA? Why is it currently sticking to DNA? Charges. So what if I could somehow like cover up these charges or neutralize them or make them negative? Then wouldn't DNA let go? So that's what we do. We have this chemical group called an acetyl group. Acetyl, ACETYL. And you don't have to know like the chemical formula, but it's this COCH3. And basically, we can add acetyl groups to the positives, and they neutralize the positives. So it's kind of like this. Pretend You know how there's two opposite sides of Velcro? They call one side positive and one side negative. One side's kind of fuzzy and the other has all these like little bitty hooks. So think of it as Velcro. Here's some positive Velcro. Here's the negative side of Velcro. If I want them to not stick anymore, what if I like just took some tape and I just covered all of this Velcro with tape? Now, this guy wouldn't stick. That's what we're doing. We're basically covering up the positives by adding acetyl groups. So I acetylate that, and I acetylate that, and that, and that, and that, and that. And eventually, there's no more positives. So now my DNA is not attracted. So now my DNA is held kind of loosely. And so now I can read those genes. Okay. So one quick little aside, because this I think will bother some students. It would have probably bothered me. I took AP Bio in the 93, 94 school year, so we didn't know any of this yet. But what looks weird about that formula, COCH3? What looks like incorrect based on 10th grade chemistry? COCH3. There should be one thing that's like, well, why are you writing it like that? Yeah, shouldn't you just, like Mr. Cotton, you don't say HHO, you say H2O. Like, why don't you just say like C2OH3? Well, in organic chemistry, you guys haven't had it. You won't have it until like generally your sophomore year if you're like a bio major or something. So when organic chemistry writes something a certain way, that also tells you how it bonds. So you don't know these rules, but this is basically saying, oh, COCH3 looks like this. Like all organic chemists agree, I take a COCH3 and then maybe I attach it to a histone right there. So like, here's my histone. So they all agree just based on the rules. So C has an O bonded to it, and then that C also has a CH3 bonded to it. So that's why they write it funny. You don't have to know that for AP Bio, but that's mainly just like, why in the heck do they sometimes do weird crap like that? So I add an acetyl group. When I add acetyl groups to the positives of histones, that neutralizes positives. Now my DNA is not held down. I can unzip it and start reading genes. You're like, okay, very mildly interesting, Cotton. In fact, I'm going to go with not very interesting. Here's what's interesting. You know what allows me to acetylate and deacetylate histones? Life experience. So when I eat the Big Mac instead of the salad, different genes get acetylated and deacetylated. It's changing my genetics. I'm not changing the ATGC, but I'm changing which genes are on and which ones are off. I've got a gene for brown eyes. I've got a gene for dark hair. I have gray hairs right here. This gray hair has a gene that says it's dark, but that gene is off, and it's going to be off forever now. That hair follicle is always going to produce gray hairs. So how different is it to have no pigment going into the hair versus a gene that says put pigment in there, but it's always off? Like if you have no gene for brown eyes, you have blue eyes. But what if you have a brown eye gene, but it's always off? Don't you have blue eyes? So isn't having a brown eye gene that's always off the same as having a no brown eye gene? So when I eat things, I change which genes are on and off by acetylating and deacetylating histones. When somebody gives me encouragement or yells at me and cuts me down and embarrasses me in front of everybody, I acetylate and deacetylate histones. It's not changing my ATs, Gs, and Cs, but it's changing who's on and off. Kind of makes sense. If I drink, again, back to the chugging a Coke and eating Snickers analogy, if I'm chugging Coca-Cola, don't I need to like turn on genes for insulin so I can make more insulin and like bring my blood sugar back down? If I'm not drinking any sugar, if I'm on like some sort of keto diet and I'm not drinking sugar or eating sugar, then shouldn't I make like less insulin? So based off of what I eat and drink, aren't I turning genes on and off? You're like, okay, no big deal. But it also works with emotions. So whatever you're feeling emotionally is also turning genes on and off. So when you're mean to that kid, you're not just mean to that kid, you're changing gene patterns. Now, some are temporary, just like diet. I ate something for breakfast with a lot of fat. I ate something with very little fat at lunch, so I change it. But some things have long lasting effects. So, what if the way I'm raised by my parents means that I never really feel secure? I never really know if they love me. We never hug. They never say love you. They never do something else. I realize some families don't hug, but it's still very evident that like mom and dad like loves you. So you're like, yeah, my dad doesn't hug, but like I know he cares about me. He shows up at practice. He supports me and all that. What if they don't? It turns out these on-off switches get passed on to your kids. So what I eat, drink, and how I feel about myself now impacts my kids and my grandkids. I am not making this up. Now, today I won't have evidence for you, but next class I'll show you the evidence of this. So remember that when you're talking to people and all that, you can joke with your friends. Like if they're not offended, kind of like when I joke with my wife, that's fine. But when you're like mean to somebody, you're turning genes on and off. Super weird, isn't it? So, we used to have any of y'all like finished a psychology class? Like maybe you took psych last year? Got one, two. Is anybody in psych this year? Three. Have you guys talked about the whole nature versus nurture debate? So in nature versus nurture, it used to be like, all right, schizophrenia. 1% of the population is schizophrenic. But if you've got a guy that's schizophrenic and he has an identical twin, there's a 50% chance that the twin is schizophrenic. So that means schizophrenic is highly genetic. So if it wasn't highly genetic, there should only be a 1% chance of this dude being schizophrenic, right? Why do I suddenly have a 50% chance? Well, my brother's a twin and he's schizophrenic. So now that would be highly genetic. So we're like, that one's kind of more nature. But then there are some people that are twins and one's schizophrenic and one's not because they grew up in a different environment. So how does that work? So in psychology, up until recent times, when I was in AP Psych my senior year, they're like, hey, sometimes it's DNA, sometimes it's how you're brought up. We don't know. Then we discovered this. Now it's how you're brought up manipulates your DNA. That's why so many things are like kind of nature and kind of nurture. I might be genetically predisposed to some sort of disease, but based off of my environment, it either happens or doesn't happen. I might have genes for something, but if I've got them on or off, they're manipulated. My environment turns things on or off. So I might have DNA that says you're supposed to be this, but based off of my environment can say no. Super weird. Okay, let's look at one more thing right here. Still looks fine on chat. I don't see anything new there. Now let's look down just a little bit further. So whenever we manipulate histones, that tends to be more transient. That doesn't tend to be like long-term, 20 years type of effects. There are effects that are more permanent. So down below that, it says epigenetics before transcription, DNA methylation. So now we're going to be in this section. Let's say that I got some DNA in here. So here's a strand of DNA. And we'll just get to introduce DNA methylation, and then we'll get into the cool stuff. Oh, yeah, my fault. I misread. I thought I was 5 minutes away. I thought I had 5 more minutes. So since I don't have 5 more minutes, we'll stop at histone acetylation. So next time we'll pick up with methylation. So if I were to like ask you, because I probably will on Monday, I'll write myself a note. But on Monday, if I'm like, where were we at? You could say, we're right here. Like we just finished DNA acetylation, or we're getting ready to start methyl, or something like that. But I'll write myself a note. So fourth, third, finished, histone acetylation. Okay, let me submit my official attendance. And then if you're on the Zoom, as always, if you have questions or concerns or anything or comments, stay on the Zoom. If not, have a wonderful weekend. We will go over the quiz on Monday. So see ya. Oh crap, I forgot to turn the volume back up like I said I would. Hope nobody was